Thank you, Beatrice, for that introduction, and thank you all for coming. I am beyond thrilled to be here. I love opportunities to come talk to students, not only about my science and the science part of my life, but also my journey. How did I get here? How did I get standing before you today coming from UF? So what I thought I would do is spend maybe just 10 or 12 minutes or so in the beginning and just kind of tell you who I am, where I came from, and how I got here. And then we'll talk about some cool science, and then a little spoiler alert. We have a NASA study, so at the very end of the talk, for about a few slides, I'll tell you a bit about the space study that we're doing, which is super exciting. So who am I? Where have I been? How did I end up here? Well, I should start with I was born in Alabama, here, Mobile, Alabama. And from there, you'll see my trajectory. I've been all over the country. So from there, I moved to New York at about 8 or 9 years old. My parents divorced. And so I went with mom to grandma's house at about that age. And then after that, time for high school. Then my dad got custody of us. So then I moved on and lived with him in Chicago, went to high school, everything great. From there, I went back to the East Coast to Howard University, which is where I started college. For those of you that aren't sure, that's at HBCU, Historically Black College and University in Washington, D.C. So I spent some time at Howard, learned a lot, partied a lot, and had a great time. Didn't get as much done, which I'll get to in a moment, but my parents quickly said, you're not really doing what you're supposed to be doing, so you're gonna have to come home. Well, at that point, they had moved to California, so home for me was LA. So I moved from DC all the way to LA to come home. And that's when I got my act together eventually, and I'll talk a little bit about that, and enrolled at CSUN, California State University, Northridge. And so that's where I finished my undergrad degree. I also got my master's degree there. And then after that, I wasn't sure what to do with myself, but I decided more education had to be in the future, so I decided to go on and get a Ph.D. And so I sent myself back to Chicago to Northwestern University. I was familiar with the area, so I felt like that was a good place to go. And then did my PhD there, stayed also for my postdoc, which is about a year or two that you do after your PhD. Kind of like the equivalency of like a residency for doctors. It's sort of that for PhDs. So I stayed there and did my postdoc, and then from there, I am now here at UF at Florida Gator. So I've been all over, haven't stayed in one place for too many years, but I'm here now, and I think this is home. So that's sort of been the journey, but really a lot happened in all of those treks across the country. A lot happened in my growth and development. And so even though these are nice smooth lines, it really wasn't that smooth. So let's look at that. So I have this nice path. Everybody's probably seen this, right? This is at the top is sort of your expected path to success, how it's going, how you're gonna get there. You're gonna hop on your journey vehicle, bike, moped, whatever, and you're gonna ride straight to your goals, whatever your goals are. You're gonna go there and it's gonna be smooth sailing, no problems. The reality is, it kind of looks more like this. All kinds of hills and valleys and things in your way that you have to figure out how to get around. It's not a clear path. And so for me, it definitely wasn't either. So I found this image. I didn't even create it, but it's crazy because when I fill it in, you're going to see it kind of fit perfectly for my life, right? So here's me starting out, and I'm kind of talking my life starting when I was your age, right? So freshmen, sophomores in college. So moving along through life, the first thing that happened is, and you probably got that already, but I dropped out of college. So you're actually looking at a college dropout right now, right? Crazy, right? So that was my first little hurdle to overcome. And then eventually I decided, of course, I've got to go back to school because I wasn't living my full potential. I wasn't doing everything that I felt I needed to be doing. So these trees here, that represents undergrad and grad school. Lots of little, things you have to kind of navigate around, right? I was a single mom at that point, so my son is in the back, so he knows because he was there the whole time. So I had Warren in between college dropout and finishing undergrad, and I was a single mom. So here I am trying to finish undergrad and go to grad school, and I'm a single mom. Very rare, not very common. I was the only student in graduate school with a child. It was just not ideal. So keep moving along, because again, I'm still trying to get here to my goal, right? And nothing's going to stop me, not even these trees and being a single mom. Somewhere along the way in grad school, I lost my mom. So devastating, right? Another huge crater in life, just a challenge. But again, I'm still trying to get to my goal. And although there's these things that are blocking my path and making my path harder, I'm still trying to get there. So, all right, that happens in grad school, so that's terrible. But I got to keep going, right? It's not really a choice. Then I get to UF, and I'm on the tenure track, and then we lost a child. Another huge gaping hole, right? Another challenge in life. Reasons to say, I'm not going to do this. I can't do this, right? But you don't. I'm still trying to get to my goal. Grant rejections during the tenure track, like a lot of them. Like most of them. Failed experiments, things don't work. You spend months and months, things just don't always work out. Difficult students, had to overcome all of that. And all of this is, you know, while at UF trying to get to my goal, which is to become a full professor at a Research 1 institution. But I made it. I made it. Right? I just recently got tenure. I put in my packet. I had an outstanding packet. I've brought in over $3 million in funding since I've been at UF. Eventually the grants obviously started to come through. Got rid of difficult students and worked with some of them and got, you know, a good team together. Still have failed experiments, but we've just kind of learned some different approaches to kind of address some of those challenges. But at the end of the day, I got it. So I share all of this incredibly personal information with you for a couple of reasons. The main reason is I just want you to see that as you're trying to get to your goal, whatever it is, graduate school, hopefully, medical school, graduate school, you want to start a company, whatever your goal is, if you're expecting this and then you get this, it can be really distracting and hard to figure out how to get back on track. But the take home is the path may be bumpy, but you still can make it through. It's not giving up. So I have a few more comments about that. All right, so that's super sad. How did I make it there? And so the most significant thing was I had great mentorship. So during my undergrad work, I worked with Dr. Maria Elena Zavala, who is a Native American and Hispanic woman. I related with her. She was an underrepresented minority person. Somehow I felt some connection to that. But she took me under her wing and, you know, mentored me to getting into going to grad school. And then I got to Northwestern and I met Guillermo Amir, who was my doctoral advisor. And he, again, cared, mentored, invested in my future to get me where I needed to be. And of course, then I get to UF, and I'm just so privileged to have also an underrepresented minority female woman that is the chair of my department, literally leading by example to say, I can do that too. I can get there. So she was Dr. Michelle Manuel, who's the chair of our department now, tremendously supportive of my career path. Some of those challenges you saw happen during the tenure track. So just tremendously supportive, always keeping me focused. What do I need to be doing to get to tenure and that kind of thing. So mentorship, great mentors along the way is really, really important. And that starts right now. Right? So while you're in undergrad, finishing undergrad, those people that have shown interest in you and I want to help you succeed, listen to them, take their advice. If you have questions and run into troubles, go find them, seek them out, and utilize them because they've been there. So. All right, so I saw this figure and I was like, this is another version of my path, but what I thought I would do is just give you some tips because, you know, likely this is the path for a lot of people. Life is unpredictable. You never know what kind of stuff you're going to be faced with. So this was my road to success. You'll notice there's lots of failure, right? I failed all kinds of times, right? But the path continues past those failures. So I always call it my path to greatness. My mom used to always say, greatness is where I was trying to get. So this is my path to greatness. So the first thing is you will get knocked down. You just will. That's just life. There's good and there's bad. You will get knocked down. The key is you have to get back up. You just can't stay down forever. When I faced all of those challenges, I could have just stayed down and, oh, this is so hard. Oh, it's just too much. But I had to get back up. I was never going to get where I wanted to be if I didn't get back up. Okay? So that's the first thing. Get back up. The second was dream big. So I was like, yeah, I could do a master's degree. I could live with that. But Dr. Zavala taught me dream bigger. Why not a doctorate? Why not? And I was like, yeah, that's a really good point. Why not? So just dream big. Whatever your dream is, that's great. But there's got to be something even beyond it. Don't hold yourself back from going even beyond in your dreams. Okay, so dream big and then go bigger. That's something I did. Believe in yourself. It's a little cliche, I know, but it's so true, right? So many times when you have those moments where you are knocked down, and we, just nature, we tend to just say, well, maybe I'm not good enough. Maybe I really can't do this. When I had all these grant failures, I thought, maybe I can't be a college professor. Maybe this just isn't what I should be doing. And really it came in there, just that idea of believing in yourself and realizing you belong here. You belong in the STEM field. All of your voices matter. All of your perspectives matter. You belong here. So that was big for me. This is a tough one, but you've got to learn to love criticism. It's so hard, right? It's not the most fun. But I say that really just kind of getting at this idea of thinking of what you can take from that criticism to make you better, right? Filter out the stuff, that's maybe not going to make you better if there's some unhelpful criticism, but always try to find different things in criticisms that you get along your way to just fuel you, make you better. So that was a big one for me because I'm very sensitive. All right, love what you do. So my mom used to always say this one too. Whatever that is, if you love it, it won't feel like work. It'll be something you want to do. And some of you are shaking your head. You've heard this expression. Again, grant failures, difficult students, so many reasons to leave, but I stayed in part because I absolutely love what I do. I just do. So, love what you do. Don't stop no matter what. This kind of goes back to the get back up. Again, life knocks you down. Circumstances knock you down, a million reasons to stay down. Life is too hard. These challenges are too hard to overcome. Don't give up no matter what. Don't stop working towards your goal. If you're in college and somehow there's some financial burdens and that kind of thing and you want to drop out, don't stop no matter what. Take one class. Take an online class if you can't get to campus or whatever, but just keep moving forward towards your goals. Think outside the box. This actually kind of applied throughout my life. I didn't see it until more of a research side of things, because I think outside the box, taking non-traditional approaches to solving my experimental challenges. So I feel like I've been doing it all along. But really, I think when I think back on my life, I've been thinking outside the box way earlier. My path to getting to a professor is non-traditional. Right? I dropped out of college, single parent. These are just not your typical characteristics of graduate students. So just thinking outside the box to how to make it work, right? How to make it work. For me, I had a child, figuring out how to navigate all of those things. So just thinking outside the box and being creative to solve your problems. And then that just goes back to my previous slide, get a great mentor. So there's thousands of these that swirl in my head all the time, but these are just the ones that resonated with me most that I wanted to share with you, sort of how I survived and made it to the end. Okay, so that was my opening part. I told you already that I am, or actually Beatrice mentioned in the introduction, I am a cell and molecular biologist. That is what I know, that is what I do. I did my Ph.D. work at Northwestern in a biomedical engineering lab. So this was like my first introduction to the fact that we can grow cells on something other than Petri dishes. I just thought that's what we did. So this was my first introduction to materials. And so this kind of goes back to thinking outside the box. Everyone else I knew, coming with the same degree I was getting, was going to biology departments or med school and that kind of thing. But I was thinking outside the box. I could do something totally different. And here I am in a materials department. I'm actually a biologist. So the work that we do in the lab is tissue engineering and regenerative medicine, which really is a nice interplay between the two. So our lab, we do cell-based strategies to address tissue engineering challenges, but we also do some material-based. So I'm going to tell you a little bit about both aspects of our research. So if you're squeamish, there's a picture that's going to pop up here. I don't know if you are all biologists? Biology people? Most? Okay, good. All right, so you shouldn't be grossed out. Okay. All right. So just by definition, just to get us all on the same page, tissue engineering, the goal is to replace or repair or restore damaged tissues in the body. So for whatever reason, a disease, maybe a birth defect, a congenital thing, there is some tissue in your body that needs replacing. So this field has been around for a very long time. I have this one example, the Vacanti mouse. In this case, this mouse, underneath the skin, they implanted a synthetic scaffold that's in the shape of an ear. And then they seeded that scaffold with chondrocytes, which are cartilage cells. And then it grew as the mouse, sort of as an incubator almost. And then you have essentially an ear that you could use. It's not functional, but it's at least usable for burn victims or people that were born with defects and didn't have these structures. So this could be a tremendous change in someone's life. So this is one example of tissue engineering. Here's another, this is an artificial trachea. So in this case, again, a scaffold made out of a material was seeded with the cells, and then the cells are allowed to grow on the scaffold and recreate this tissue. So this was actually implanted in a little girl, I can't remember the year. It must be 2013, was implanted in this little girl at 2 years old. She was born without one. She was able to get this implant, and the cells they used in this scaffold were her own stem cells. So this was groundbreaking work, especially particularly because they put it inside of a patient, and it was successful. The graft was successful. And then we have skin substitutes as a third example of tissue engineering. And again, in this case, you have sheets of material that are seeded with fibroblast cells, which are the cells of your skin, and those are grown to create skin substitutes. Again, used for burn victims and patients that need skin coverage. So this is kind of the idea of tissue engineering. We're trying to engineer tissues for diseased or damaged parts of the body. So that's what our lab does. So if we think about tissue engineering, the kind of general schematic is you've got a patient with some tissue that needs replacing, whatever it is. There will be some cells that you're going to get from the patient. Those cells will be combined with a biomaterial. That biomaterial and cells combination will be grown in the lab under some well-defined conditions to recreate the tissue, and then it'll be reimplanted in the person. And so this looks really, really simple, but there are challenges at every single step of the way. Like when we think about the cells, where should we get these cells from? What would their phenotype look like? Do they function properly? Cells do things, right? How stable are they? Can we control them? When it comes to the biomaterial, we also have challenges there. We have to consider things like, do we use natural materials like proteins? Do we use synthetic materials? How about bioactive materials that actually have some function, can actually do something? So things to think about there. Architecture, porous, fibrous, you know. And then, of course, even when it relates to the culture conditions, do we mimic, when we get the cells and the material in the lab, do we mimic healthy conditions? Do we mimic disease state? And what about extreme conditions? So a lot of things. And our lab pretty much works on all those areas. We're very broad. So, so just to kind of demonstrate what our lab is working on, we're doing vascular tissue engineering, which is the project I'm going to talk about today. That's your blood vessels. Okay, and I'll talk about that in a moment. We also work in orthopedic tissue engineering, which is the engineering of bone. So that's sort of our soft and our hard tissues. We also have some dermal tissue engineering. We're most interested in diabetic wound healing. We also do stem cell engineering. We think stem cells are a great source for tissue engineering, but we really need to have better ways to control them. So we work on just cell biology ways to control stem cells. And then finally, we do some device engineering. Our lab is working in the area of stents, which are vascular devices. So we're working on degradable ones, drug delivery ones, and other things like that. So we're kind of all over the place, but all with the overarching theme of tissue engineering. All right, so just a little introduction. So I'm going to talk about, for this talk today, the vasculature, because that's probably the project we've taken the farthest. So the vasculature is a network of blood vessels all throughout your body. So these are your arteries, veins, capillaries, everything that is circulating blood is your vasculature, right? So that network. So it's obviously critical to the health of your organs and tissues because it circulates blood, right? But sometimes segments of the vasculature can become diseased, little areas along. And that's called cardiovascular disease. So it turns out that 1 in 3 people will die from cardiovascular disease. That is staggering. 30% of deaths are a result of one form or more of cardiovascular disease. It's crazy. So disease in the coronary artery, peripheral artery, there's a couple of vessels that are particularly susceptible and can lead to death, such as those, but hundreds of thousands of surgeries performed annually to try to address these challenges. Staggering economic impact. Billions and billions and billions of dollars on care for, prevention of, surgeries to treat cardiovascular disease. So it's a big deal. It's a big problem. So we've taken it on. So one of those diseases is called Otosclerosis. And so this is where the vessel becomes narrowed by this buildup of plaque and cell debris and fat and cholesterol that builds up in your arteries. This is progressive over time, and this is just a cross-sectional view. And you can see if you're trying to pass blood through this, having it narrow off like this is going to be a problem, right? And it's going to lead to poor circulation, and it ultimately can lead to amputations, heart attacks, you name it. So it's a bad thing. So if you catch it early, what surgeons do is what's called balloon angioplasty. So they'll insert a balloon where that plaque is and just inflate it and just mash the plaque up against the wall and just restore blood flow. Stenting, I mentioned that we work in the area of stenting, is another one. This is a wire mesh that can be deployed right at the site where that plaque is, and essentially it opens up the wall and again restores blood flow. The goal is keep blood flowing, right? In severe cases, you have to do what's called a bypass graft. Almost everybody knows somebody or somebody's grandmother or a great aunt that's had a bypass surgery or a triple bypass. You've heard this expression? Okay. In this case, the goal is redirect blood around the blockage because we can't do any of these. The damage is too severe. So if this is your blocked area, just redirect blood flow around it, okay? So in the case of a bypass surgery, we have to use some vessel as that new conduit to redirect blood flow. So typically they'll take and harvest a vein from somewhere else in your body. And so the problem is 30% of patients that need bypass surgeries don't have a vessel available for you to harvest. And that could be either it was already harvested some years earlier, maybe your other vessels also have cardiovascular disease. And so it's a challenge. So we've got a patient population, 30%, that needs something else. There is something else. So it turns out we use EPTFE, expanded polytetrafluoroethylene, Teflon. It basically, the company that makes these makes your ski jackets, Gore. They make I see shaking heads, you know what I'm talking about. But it turns out what they did was they took the same material, this Teflon, and when they heated it up and they pulled it very rapidly, it separated out into this microarchitecture that made it ideal for a vascular graft. So it was actually kind of a cool little story of how Gore developed their vascular graft. But Gore's been making these vascular grafts for years, and right now they're kind of Gore and Bard, there's one or two other companies. They sort of own that market. So they make these vascular grafts out of Teflon, but they're not perfect, right? They're what we're using. This is the current standard of care. None of you would need it, but if anyone much older needed it, this is what they would get for a bypass. And the problem is two things. These can cause blood clots, and of course, if you're causing a blood clot in this vascular graft, that can lead to heart attack, stroke, all kinds of things. The other thing is something called neo-intimal hyperplasia. Long word, but essentially what it means is smooth muscle cells, which we'll talk about in a moment, they migrate into the vessel, the graft, and they just grow and grow and grow and grow, and eventually they start to occlude or block the vessel too. So in all cases, the synthetic graft is currently used, but over time, somewhere around the 5-7 year mark, they start to fail. So the surgeon has to go in and place a new one. So this is kind of a revolving bad situation. So this motivates the field of tissue engineering because can't we make it? Can't we make a suitable blood vessel replacement that could be used instead of the synthetic non-cellular material? So that's the motivation. So in our lab, we're interested in three main research thrusts: cell engineering, biomaterials engineering, and then dynamic environments. That's the space study that I'll talk about at the end. So here is our approach to tissue engineering. First, let me tell you a little bit about blood vessels, because we're trying to mimic a blood vessel and make a blood vessel, we should understand it. So this is a cross-section of a blood vessel. There's a lot going on there, but the two most important things in the blood vessel are your endothelial cells and your smooth muscle cells. Now, your endothelial cells, they line the inside of the blood vessel. So these cells are critically important. They essentially keep your blood fluid, is sort of the expression. Without them, your blood would be clotting and it would be terrible. So your endothelial cells, they secrete small factors into your blood that basically is an anticoagulant and keeps your blood liquid, keeps it moving, right? Smooth muscle cells, on the other hand, they're in the wall of your blood vessels, and they essentially keep the vessel tone, keep your blood vessel pulsating like this to keep blood flowing through. So these two cell types are going to be critical if we're going to try to engineer a blood vessel. So keep those two in mind, endothelial cells and smooth muscle cells. So I mentioned endothelial cells. So we actually in our lab are very interested in stem cells and progenitor cells. And so the cells that we use primarily are called endothelial progenitor cells. And they're just like the name says, they're the progenitor cells to the endothelial cells. So they're a little more primitive. They don't have the same potential as a true stem cell, but they have multiple potentials. So we kind of are interested in this population. So these cells we are interested in because we can get them from whole blood. So it's pretty non-invasive to isolate these cells. They're in circulation, and we think that they could be a suitable source to make our vascular graft. So a little bit of the data. So I mentioned we get these cells from blood. So this is just showing how we got the cells. We get the blood, we spin it out, centrifuge it, and then we get this what's called a buffy coat, basically a little dense layer of cells. If we take those cells out and culture them on fibronectin, it's beautiful. After about a week, you get these nice stem cell colonies or progenitor cell colonies. And it's those colonies that came out of blood that we just let them continue to grow, and they grow beautifully, that we use for our vascular grafts. So they look like endothelial cells at this point, but of course we're not going to use them unless we confirm. So we did immunohistochemistry. So this is staining for markers on the cells that would be there if they're endothelial cells. And we're getting positive marker expression for those cells using immunohistochemistry. We also did flow cytometry to essentially assess the whole entire population, just making sure that it's a homogeneous population of endothelial cells. And looking at the same exact markers, we're getting full expression of the population for those markers. So we're feeling pretty good. The cells look like endothelial cells, and they stain for markers that are specific for endothelial cells. So far we're thinking that we're pretty good for a cell source. So the next thing is all of the cells in your body have some function. They do something. They don't just sit there, right? And so endothelial cells, I mentioned earlier that they're critically important in keeping blood liquid, essentially. And they do that because they secrete small molecules. And so it's not so important which molecules, just to know that the progenitor cells that we isolated do secrete those same molecules. So now we have cells that look like endothelial cells. They stain, have markers for endothelial cells, and they function as endothelial cells. So we're off to the races. We're very happy. We've got a nice viable cell source for our vascular tissue engineering. Of course, can't all be great. The problem we found was that it took somewhere on the order of 1 to 2 months to actually get those cells to differentiate. Now, imagine you're a patient and you need a vascular graft. You're coming into the clinician, you're not coming 2 months before you need this vascular graft. So this is a challenge. We need to figure out better ways to control these stem cells or progenitor cells. 30, 50 days is just not going to be a viable option. So that's what I just said. We need better ways to control these cells. All right, so that leads us to this idea of cell engineering. How can we engineer cells to do what we want them to do? So you guys are all biologists mostly, so you understand the concept of receptors on the surface of cells? Good. So you know that receptors on the cells bind to their ligand, and that activates a whole process, a signal cascade that causes cells to do things. So I have here, so you activate your molecular pathway, which controls gene transcription, which can control the cells differentiating, growing, dying, migrating, whatever it is, the process, right? So we decided, let's go after that strategy. Let's target some specific receptors on cells that we could use to promote these cells to differentiate. And maybe we could get that differentiation to happen faster than 2 months, was one of our thinking. So we've targeted what's called vascular endothelial growth factor receptor, VEGFR2. There's a 2 at the end. This is a receptor that binds VEGF, vascular endothelial growth factor. So when VEGF binds VEGFR2, you get activation and cells differentiate, they migrate, they do all kinds of things that ECs, endothelial cells, should do. So we decided to target that receptor. And so I just kind of have a little bit just showing how VEGF receptors, there's 3 of them. Different VEGF can bind to the different receptors. There's all this sort of non-specific nature with which VEGF binds the receptor. So we knew if we want to target this receptor, we don't want to use the growth factor because it's not specific, and there's all this off-target binding. We needed something specific and purposeful, and that we knew would bind VEGFR2, and then we could promote this differentiation. That was our rationale. So that's our goal. Develop a VEGFR2 agonist, receptor agonist. We needed it to be stable, targeted. We need to be able to control it. And we can use it soluble by adding it to culture media, but we thought, wouldn't it be nice if we can immobilize it on our biomaterials? So then maybe we eliminate the step of having to culture the cells ahead of time. So we had some criterion. So what we did was we turned to using DNA aptamers. And before this project, I had never heard of aptamers. Through all of my biology training, I had never heard of aptamers. So aptamers are single strands of DNA, and they're very short, somewhere around 20, 40, maybe even 80 nucleotides. So the cool thing about them is it's this single strand of DNA. They fold into these really cool tertiary structures. You get hairpins and loops and so forth. And when they fold into these cool tertiary structures, they bind to whatever their target is. And with aptamers, the field has been really, really well used. They're used for all kinds of bindings. But they bind to their target with amazing specificity, targeting, affinity, much better than antibodies, much better than growth factors. And the key then for us was, okay, they bind, but do they activate? And so that really became our next challenge. This is just a few things aptamers are used for right now. Drug treatments, imaging, diagnostics, things like that. This is a resolution. If you actually looked at the structure of the protein aptamer binding, they kind of just fit into a little pocket or a cleft on the protein. So the binding is more in intermolecular forces, hydrostatic interactions, and things like that. So here was our idea. We found a company that sells a VEGFR2 binding aptamer. Great, we don't have to develop it. Fantastic. So it's 80 nucleotides. We predict that it folds into that structure, this big loop in the middle and a couple of loops on the ends. And so we have that. Now, it's not known to activate the receptor. We just know it binds the receptor. And ultimately, we want activation because we're trying to get some cell processes downstream. So we look at VEGFR2, the receptor, and it's actually composed of 2 dimers. Right? So we thought, what if we took 2 of these monomers, we call them monomers, tether them together with this hexaethylene glycol 18-atom spacer, and create what we termed the divalent aptamer assembly. It's essentially 2 monomers tethered together. Pretty straightforward. So that was our idea. Sorry, I should have popped those up. And so here's sort of a cartoon of what that might look like. In soluble form, we've got these little divalent assemblies floating around. Here's our inactive VEGF, dimers open. Hopefully, these the divalent aptamer will bind to the dimers, somehow cause a conformational change or dimerization of that receptor, and then we'll get downstream cell processes. So this was what we were hypothesizing would happen. And it turns out we were right. So here's just a little schematic. This is VEGF receptor showing the dimer binding to VEGF. And this is just showing some pathways that become activated. So the first thing we wanted to do, we know the first step in receptor activation is phosphorylation, right? So we just did a phosphorylation assay. Do we at least get that? Can we start there? And we were really excited to show, this is our aptamer in orange, that our divalent aptamer does actually cause phosphorylation. So we're pretty happy. The next thing we did was look at some of these different pathways, and you guys know as biologists, molecular pathways, there's tons of them and they overlap and it's super complex. But there's a couple of them that are pretty key, like the AKT pathway. So we did flow cytometry and we confirmed that our aptamer, our divalent, actually causes activation of AKT pathway. So we're pretty excited. Then we decided, so we're kind of jumping along because we want to kind of get to the data. Is this promising before we spend too much time on it? We know that one of the things endothelial cells do is make nitric oxide synthase. So we wanted to see, does our divalent aptamer promote the production of nitric oxide synthase? And we saw that it does. It's a small increase, but it was still there. So we were very excited. So these are the molecular events, some of them, that our divalent aptamer was able to initiate. We then needed function function, because you know I'm big on function. Just because it looks like an EC doesn't mean it functions like an EC. ECs are endothelial cells. So we did a scratch assay. And in this case, you grow the cells till they're confluent, completely covering, and you literally scratch and create a clearing. And then you come back the next day and see, did the cells migrate in and fill in that cleared space? And so we found that when they were incubated with our aptamer, they did that. So our aptamer is able to promote cells to migrate. Then we looked at tube formation. These are endothelial cells, so they should assemble like vasculature. And so we saw that this is VEGF, here's our aptamer, that we were able to get some assembly of the cells when they're grown in a hydrogel. And this is just quantifying tube length. So we're able to show functionally that the aptamer also causes the cells to assemble in like tube-like structures. And so the last piece of data that I wanted to show is the goal here is to show that this aptamer assembly that we've made has these biological processes. And mind you, this hadn't been shown before. These people, the company that was making this aptamer was only selling the single aptamer for binding. It wasn't selling it as a divalent and for activation. So we were kind of in uncharted territory. So this is a chick embryo assay. You take fertilized chicken eggs, and we took filter paper, that's what the dotted line is, and we soaked it in either the aptamer or saline or the growth factor. And we saw this beautiful blood vessels forming. So we show that our aptamer is actually able to promote vascularization. Huge when it comes to tissue engineering. So now, actually this is not as up to date. We have actually shown that we can promote differentiation. We started with adipose-derived stem cells, which are stem cells found in fat tissue, but we did show we could do that. We're also trying to modify the aptamer. Right now we just have the two together, but what happens if you put 3 or 4? And we have this 18-atom spacer, but what happens if we make that spacer longer or shorter? So we have some chemistry here that we're kind of trying to play with a little bit and just kind of see what the potential is. We're also trying to deliver it differently. Right now we just put it in the media, but can we immobilize it? Can we deliver it from nanoparticles? And then finally, we really want to resolve where is the aptamer binding to that receptor. In my little cartoon, it looks like the divalent is grabbing both of the subunits, but is that really happening? Where is it binding? So we have a bunch of work. This is a very active area of our research group, this sort of small molecule development. All right. Okay, good. So I'm just going to talk briefly about the biomaterial, because I've told you the cells, and I've shown you that we have this interesting approach to how to get endothelial cells, which are the cells you need for the vasculature. But let me tell you a little bit about our material. So this is going to be a blood vessel, right? And we're going to put cells on it, but until the cells make tissue, this blood vessel has to be the same as the material scaffold has to be the same as a blood vessel mechanically. So it's got to be durable, flexible. We have to be able to suture it in. And I list that. People always ask me, why do I list it? Because it's really not trivial. Being able to suture something, once you create a hole with the needle, it's a defect in the material, and it could be a point of failure. So that's a pretty challenge there. It's got to have the same compliance or mechanical properties to the host tissue, native vessels. We have to think about things like burst pressure. It can't kink when it bends. It's got to stay open. From a biological perspective, we've got to pick a material that's biocompatible. It's in contact with blood, so it's got to be hemocompatible. It's got to support the cells. We think it should be biodegradable, with the goal that the cells will eventually make new blood vessel tissue, non-toxic, all of these things. So we have a laundry list of requirements for our material. And we have a couple of approaches. So what we do is we're interested in mimicking native blood vessels. We kind of think of it as being biomimetic. So this is a cross-section of a native blood vessel. And you can see sort of the squiggly lines. Essentially, the blood vessel is composed of nanofibers of collagen and elastin. That's what gives your blood vessel its elasticity. So we decided, well, we can make nanofibers, so we use electrospinning. We electrospin collagen and elastin because those are the components in a blood vessel, but we also added in a polymer component, which I'll talk about in a minute. This is mostly talking about our synthesis. So we have collagen, we have elastin, and we have our polymer. And this is the electrospinning to create the nanofibers. And so the goal is ultimately to make a tubular scaffold that looks like a blood vessel. So I'll tell you about our polymer. And I am not a chemistry person, so this is a very, very simple polymer. It's made out of citric acid and an aliphatic diol. And so the citric acid, you guys all know, is a part of the Krebs cycle. So making a material out of citric acid is not bad. The aliphatic diol essentially just means it's a long carbon chain, and it's got a couple of functional groups at the end. And so essentially, when you add citric acid and the diol, you create this network. It's just like a chain link fence almost, right? And because of that architecture, like a chain link fence, it can stretch and recoil. So it's a very elastic material. So here it is, shown here. This is a tube, a sheet, a porous tube. These are SEM images just showing the pores that we can create, but this is really a cool image showing the elasticity. So here we're doing a tensile test where we're clamping the material and then pulling it in tension until it fails. And if you look at this picture versus this, you can see where it failed, but you see that it's, when you put the pieces back together, it's the exact same size. So we get 100% recoil back to the same size. It doesn't deform and mess up. So this is an elastomeric material. So this is perfect for blood vessels. So we were able to make the tubes that are like blood vessels. We ended up making our design 2 phases. We have a solid inner and a porous outer. This is just showing the fibrous nature that I mentioned. We quantified fiber size. We've got a nice distribution. We looked at how hydrophilic it is, water-loving, because cells prefer hydrophilic surfaces, good hydrophilicity. Most important was our mechanical testing. You have to be able to make a material that's going to be mechanically robust. Can you imagine putting it in the patient and it bursts? So it's got to be able to withstand physiological burst pressures. It's got to stretch and contract without permanently, stretching out. And so here's some of our scaffold relative to native arteries, saphenous veins, and other materials. And what's really important is the compliance here. That essentially talks about the degree of deformation you get under physiological blood pressures, 120/80. So relative to native tissues, we're right on par. Burst pressure is very high, 2,600 millimeters of mercury, so definitely can withstand 120/80. And the rest of this mostly talks about the material properties. And of course, we do show that cells like it, so it's biocompatible. These are our endothelial cells. These are smooth muscle cells. So we show that both cell types like our material. Both cell types grow on our material, which is going to be important. And it's non-toxic. So this is the fiber layer, the POC, and this is just tissue culture polystyrene as a reference material. All right, so just to wrap up that, and for the last just 5 minutes, I'll just tell you, do we have 5? Okay, about the space study, because that's cool. So hopefully you were with me still, but I showed you some data about using aptamers as receptor agonists, a new function for those, for aptamers that hadn't been reported before our group. But we think it kind of opens up the door to a lot more than even what we're doing. And also I showed you this promising hybrid scaffold approach to creating blood vessels. All right, so let's talk about dynamic environments. Before I go there, I will tell you, once we create this, where we have the material, we've got the cells, we do actually grow. We have a lot of bioreactors in the lab, and so we have one that exposes the scaffold to flowing fluid to mimic blood flow, and also to pressure, so it stretches the vessel. So we are actually, when I say dynamic environments, we actually do study our systems under what would be physiological for the body. For example, also in our bone studies, we do compression to kind of mimic walking on bone. So we do try to mimic in the lab what the tissue would be, what exposure the tissue would be under. Oh, there it is. There's a picture of the one for the blood vessel. So the vessel that we would create would go here. Media would flow through it to create shear stress, like blood, and then it's pressurized. Okay, let's talk about space. All right. Totally shifting gears, but this goes back to that idea of thinking outside the box. I literally knew nothing about space before this project, but I saw a call. They were looking for junior faculty people who were new to space biology, and I was like, well, I'm new to it. I'm a biologist. I can handle this. So I wrote a grant to look at space environment, the space extremeness on stem cells, vascular stem cells, it turns out. So we know in space the 2 main factors that are around is microgravity and radiation, okay? When an astronaut is in space under these conditions of microgravity or radiation exposure, all kinds of systems in the body are affected, right? bone and skeletal, the sensory motor, muscle, sleep cycle. There's actually more. This is just a few. But it turns out the cardiovascular system is also altered from being in this environment, particularly for deep space missions where they're out for much longer. So that was kind of my link. Remember thinking outside the box? I was like, I know cardiovascular disease, so I think I could work this. So if we look at what happens to the body in the space environment, this is normal on Earth, 1g of gravity. When the astronaut, human body, whatever, goes up into space, you can see there's this shift. See how the legs have thinned out, and see how the top of the body is larger? Essentially, everything kind of pushes upward, right? And you can see the heart, this picture doesn't show it great, but the heart works harder in space. And so it turns out you get what's called cardiovascular deconditioning, or I should say astronauts get. And so they get orthostatic hypertension, there's changes in blood flow, changes in blood volume, so a lot of changes to their system. And it turns out that this essentially going to space disrupts vascular homeostasis. The comfortableness and of the vasculature is disturbed from being in space. And that disruption can lead to cardiovascular disease. Astronauts who are otherwise healthy people have a high incidence of cardiovascular disease downstream of spaceflight. So we hypothesized, remember those repair cells I told you about, endothelial progenitor cells? We hypothesized, is it possible that the space environment is damaging those stem cells or progenitor cells, and now those cells aren't able to function properly? So that was just our hypothesis for our first grant. So we have a bioreactor in the lab. Essentially, it looks like this. It has these detachable chambers. We grow the cells in here. These connect here, and they essentially rotate. And if you look at standard cell culture, when you seed cells after about 5 minutes, 30 minutes, 5 hours, progressively the cells settle to the surface, attach, spread, assuming adherent cells, by the way. And this is what that would look like. Well, in this bioreactor, it looks like this. So within 5 minutes, of course, they're still just kind of floating. Eventually, they find each other, and they create these spheroids of cells, big clusters of cells, right? And so that happens in the bioreactor. So this is the bioreactor that we use, and it was developed by NASA, and it was specifically designed to reduce the gravitational force from 1G to micro levels of G. So we call it microgravity. Okay, so this is the bioreactor we're going to use. We're going to study our endothelial progenitor cells and ask some questions. So we had two different setups for this experiment. The first was, what if we expose them to microgravity as progenitor cells? Meaning we isolate them and then stick them right in that bioreactor and give them that microgravity exposure. So here's the setup again to collect blood. We grew them out and got these colonies after their gravitational exposure, and we did 6, 12, and 18 hours. And of course, the blue line is normal gravity. And the first thing we found is you got less colonies, less differentiation the longer you expose them to microgravity. So that automatically told us just the presence of microgravity affects the ability of these cells to differentiate. We also looked at a couple of functions, also showing that microgravity affects, but there seemed to be some repair or recoil back to normal levels over time. So your body has, your cells have repair mechanisms, basically. The second setup was, let's first differentiate them under normal gravity and then expose them. So in this case, we used microcarrier beads that had the cells on them, exposed them into the bioreactor in microgravity, and again, looked at different functions of the cells. And in all cases, we were able to show that microgravity definitely alters the cells, it alters their functions and their differentiation potential. So we learned a little bit from that first grant. Then we decided we're going to actually put in an application to send some cells to space, because this bioreactor is great, but it's not the same as being in space, right? There's all kinds of different things going on. So we are actually scheduled to be on SpaceX 14, which is scheduled for, that should say April. We got pushed. April 2nd now of this year. And so ISS, the International Space Station, is maintained about 250, 270 miles above Earth. So that's where our cells are going to eventually go. We're going to send endothelial cells, we're going to send smooth muscle cells, and we're also going to send, for the first time, endothelial progenitor cells. No one has sent those yet to space. I'm not actually sure smooth muscle cells either. So this is what that would look like. We'll prepare the cells on the beads and get them all ready to go. They'll launch. It takes about 8 to 12 minutes to get to orbit. Then they orbit for 2 or 3 days while they're waiting to line up to dock with ISS. After 2 or 3 days, they will dock, and then they'll be unloaded, and that's the start of our experiment. So you can see there's a lot more that happens in a real space study than we can mimic on Earth. There's temperature fluctuations, there's hypogravity, that 8 minutes getting up to orbit. Okay, so yes, and our mission is 3 to 4 weeks for this particular flight. This is the hardware we're using to grow the cells in. I just wanted to show you just because it looks so simple, but it actually is really neat because it's got 3 chambers that will, with pistons that'll automatically, this is fully automatic. The astronauts won't have to touch our experiment, but the pistons will push down to inject media into where the cells are so we can refresh the media through the time that they're in space. So this is, these cost about $60,000 each. I only got 3 of them, so it's a small study. But anyway, I just wanted you to see that. Just from an engineering perspective, it's kind of cool. And again, this is fully automated, so all the astronauts have to do is at the respective time point, remove this and put it in the freezer. We have RNA later and some other things that'll fix them for when they get back. So this is kind of the walkthrough. This is the dragon at the tip here, okay? I'm lost for words, sorry. So then there's the launch with the dragon at the top. Then the dragon is released at orbit, and this is where it's going to be. It sits in orbit for 3 to 5 days until it connects, or 2 to 3 days, until it connects with ISS. So that's what's happening here. Here it is docked with ISS, the dragon. This is where all the experiments are going to go. And then the hatch is opened on the top, and the astronauts will unload all of the experiments. So up until now, it's just sitting in orbit for 2 or 3 days. No temperature control, nobody's looking at it, touching it. Once it gets up here, then the astronauts can plug it in and get the 37 degrees going again. And this is just a picture of the astronaut on space working one of the experiments. So this is kind of what it would look like if all things go well. And then when it returns back home, we're going to do some transcriptomics and genomics and look at what happened and what changed. Okay, so just to wrap up, the overarching theme of our group is tissue engineering, regenerative medicine. We're looking at biochemical cues to control cells, biomaterial cues, nanofibers, et cetera, but also environmental cues. And I showed you the space environment and that. But we also look at shear stress and electrical stimuli and other things, with the goal to see how best to engineer these replacement tissues. So with that, I'll wrap up. This is my group. These are my graduate students in the lab. I did not do any of this work that was presented. They all did the actual work. I have undergraduate students, including your very own Zion, who is a SF2UF, did I write it right? Yes, SF2UF student who's working in my lab. And of course, we've gotten funding from a nice broad array of agencies to fund a lot of the work. The work that I showed today was from the US Army, the vascular work, and of course the NASA work is the space flight. So with that, I will take questions. Thank you. Thank you. Yes.